Welcome to C&EN Uncovered. I'm Craig Bettenhausen. C&EN Uncovered is a podcast series from Stereochemistry. In each episode, we'll take another look at a recent cover story in chemical and engineering news and hear from C&EN reporters about striking moments from their reporting, their biggest takeaways, and what got left on the cutting room floor. Every human who has medical intervention has benefited from horseshoe crabs. That's right. For decades, the fossil-looking, blue-blooded arthropod has played an integral role in endotoxin testing. But all that could change as US-based pharma companies continue transitioning to a synthesized compound to detect glycolipids. In a story that appeared in the October 28th print issue of C&EN, today's guest looked at the long-running fight by environmental groups, ecological researchers, and drug companies to push synthetic endotoxin assays into the mainstream and horseshoe crab blood out. We'll put a link to the story in today's show notes. I'm here with C&EN assistant editor Laurel Oldach, who wrote the article. Hello, Laurel.
Hi, Craig. How are you?
Doing all right. So for anyone who hasn't had a chance to read the article yet, can you give a quick recap of what it's about?
Sure. So this is a story that really goes into the behind-the-scenes regulatory process and what makes it so darn hard for pharmaceutical regulators to move from one technology to another technology. The doubts that are built into any kind of change and the incumbent advantage that the existing tests that are based on horseshoe crabs have over tests that environmental scientists and activists argue are much friendlier to a variety of different environments. So horseshoe crabs are a keystone species in many of the estuaries where they are found. And there's a lot of people out there saying, hey, to support the animals that rely on horseshoe crabs for a variety of different reasons, we should really make this change from wild-caught horseshoe crab proteins to cloned horseshoe crab proteins to do this important pharmaceutical quality test, especially as with the onset of more and more biological drugs, the number of these tests required gets larger and larger year over year.
And the keystone species, if I'm remembering right, you lose that species and the whole ecology of that area falls apart.
Whole ecosystem goes to pot. Yep.
So how did you come across this story?
This was a story that came to me, and I was really delighted that it did, actually. So a chemist named Tim Cernak approached me. Tim had recently been to a meeting in New Jersey where this group of environmentalists called the Horseshoe Crab Recovery Coalition invited a whole bunch of pharmaceutical executives to this beach house on the Delaware Bay during spawning season, which corresponds with a full moon in the spring. Nice. And these environmentalists really wanted to make the case that not only should pharmaceutical executives choose recombinant endotoxin tests because it's good for ecosystems, but also because it's lower risk in terms of the supply chain. So there are places in the world, not in the US, but elsewhere in the world, horseshoe crabs have been declared endangered, and that has dramatically changed the availability of their blood for pharmaceutical quality testing. The story doesn't really go into this, but it's now much more difficult to obtain the Asian horseshoe crab Tachypleus, its blood for endotoxin testing, and that's making a difference to what kinds of endotoxin tests are available from manufacturers in China.
So horseshoe crab blood, the central substance in your article, is a pale blue. It looks like a blueberry smoothie. What can you tell me about the O2 carrying chemistry and how that differs from human blood?
First of all, I love that analogy, and I'm kind of craving a blueberry smoothie now. Right. There are actually a lot of ocean creatures that have blue blood, so like octopuses, mollusks, blue crabs. I think with horseshoe crabs, it just gets discussed more because unlike all those other animals, we harvest the blood and you have a lot of photographs of it. Anyway, the blue color comes from this protein called hemocyanin, which is a protein with a heme cofactor. So heme is this highly conjugated porphyrin ring with 4 nitrogens sort of sticking into the middle of the ring that can coordinate a metal cation. And then that whole package, the heme, the metal cation, all can be bound into a protein where it can do stuff that is hard to do with just amino acid chemistry. So in humans, our heme uses iron-containing heme, to bind to oxygen. And when the iron ion is oxidized, that turns our blood red. In horseshoe crabs and other mollusks, octopuses, all those animals that I mentioned, it's the hemocyanin protein. Instead of iron in that sort of central spot in the heme ring, it uses copper. So when the copper is oxidized, that turns blue. And so that's the chemistry behind the blue blood color. One funny fact that I learned is that it actually has nothing to do with this other crazy chemistry that horseshoe crabs are doing, which is the protease cascade that they use to sense signature molecules from bacteria. And that's what we really use to test whether drugs are safe or not. Huh. But it has nothing to do with blue. And the lysate reagent that people use is not blue either. The copper hemocyanin is totally purified out in the course of producing this reagent.
So this is a very specific sounding test, this assay. How is it discovered, and why is this test so important?
Yeah, so its discovery goes back to the 1960s when there was a scientist doing sort of a summer research project on bacterial infection at Cold Spring Harbor Lab, and he happened to be using these horseshoe crabs, I think, as a model system because they were relatively easy to come by. But he found that they were kind of a terrible model system because as soon as they were exposed to bacteria, their blood clotted very quickly, and that made it difficult to work with. But in the subsequent decade or two, it turned out to be really useful for the pharmaceutical industry, which at the time was testing all of its injectable drugs by injecting them into rabbits and checking for a temperature change in the rabbits. And that was a pretty problematic assay from an animal rights perspective, first of all, because our ethical systems tend to treat mammals as kind of more important, but also because in order to prevent a rabbit from developing an adaptive immune response to your drug product, say you're injecting like human insulin, you don't want an immune response to the insulin to kind of overcome the presence or absence of these bacterial contaminants. They only tested each rabbit once. So it involved a lot of sacrificed rabbits per batch of drug product. This is an important test for pharma because basically to prevent injected drugs from giving you sepsis. So your immune system, like the horseshoe crabs, is really, really good at recognizing signature molecules from bacteria. And even if the bacteria are not present, even if a drug has been sterile filtered to remove all viable microorganisms, there's nothing in there that will grow anymore, it may still contain these lipopolysaccharides, often called endotoxins in a pharmaceutical context, that can, if they're injected, give you high fevers, headaches, sepsis-like symptoms, can even at high enough dosages lead to death. This is a vanishingly rare phenomenon today because the pharmaceutical industry is so highly regulated and because these days not only are final drug products tested, but also all of the ingredients that go into drug products, all of the water in facilities, all of the containers that touch drug products. If a product is made by culturing cells, like the media that the cells are cultured in, the dishes that the cells are grown in, et cetera, et cetera. Like, there is endotoxin testing happening at every single stage in the drug production process.
So I'm afraid I have to ask about the harvesting process. How do you bleed a horseshoe crab?
Well, it's tough because it is completely encased in this shell, right? So fishermen collect there, and there's, I think, a fairly small number of fishermen that get fishing licenses to do this kind of fishing. It's regulated state by state. But so fishermen in various different states collect the horseshoe crabs. They ship them to holding ponds outside of sort of collection lab facilities. So there the crabs are disinfected, which is important, as you can imagine, because if you have this really exquisitely sensitive endotoxin testing reagent in their blood and it like comes outside of their body and immediately detects all the like marine microorganisms that were sticking to them, you would have no longer a sensor. You would just have like a switch that has been flipped. Technicians then take them and sort of fold. So if you can imagine the crab's shell connects up with its tail. So they sort of fold it so that that membrane where the shell-encased tail connects with the bulk of the shell is kind of exposed. And they stick, to my understanding, they stick basically just a syringe into that joint and let the crab bleed for about 8 minutes. And that 8-minute bleed is something that's done to improve survivability. I think it used to be that you would drain the blood for longer. And I think that that's still the practice in some parts of the world where horseshoe crabs, again, are endangered. And so that's not ideal. After it's been bled, the crab is re-released into the wild. And there's a really wide range of estimates of mortality. Birders will tell you that 30% of the crabs that are bled die. Companies that produce this lysate reagent will tell you that 4 or 5% of the crabs that are bled die. I think it's pretty hard to get ground truth on that, as with many other things about this biochemical system, which is interesting as someone who's accustomed to just like research producing facts that everybody can agree on. So meanwhile, the crabs are re-released and they either survive or not. Meanwhile, the blood is processed to extract the protease cascade that's responsible for the clotting that was originally observed back in the 60s. There's often some sort of mixing that happens from a different number of crabs into batches. One of the recombinant reagent companies, Biomérieux, is French, and representative that I spoke to from that company sort of just compared it to like mixing champagne, where you take, some grapes from this vintage that are high in this desirable chemical and some grapes from that vintage that are higher in that, and you kind of mix them all together and produce a lyophilized protein mixture that is then can be shipped out to pharmaceutical labs all over and reconstituted. And one thing that you hear a lot from sort of microbiologists in pharma is that there's quite a lot of variability between batches. So that's something that I guess happens with blood products.
So I want to bring out something that you talked about a little in the story, and that is this: there's a differential treatment between the recombinant protein relative to the extracted protein. And correct me if I'm misunderstanding it, but as it has been, even with the change in regulation that your story talks about, the version that doesn't hurt horseshoe crabs is required to pass some purity and potency tests that the blood version does not. Is that right?
So a small correction there.
Okay.
That was something that was proposed, and that was both something that the committee of microbiologists that were making rules for the USP for many years leading up to 2022, something that they really advocated for. But they got a lot of pushback when they sort of suggested in a draft chapter of the Pharmacopoeia that this is the way that recombinant tests should be regulated. What they proposed is basically the FDA regulates horseshoe crab blood product as a blood product because that's what it is.
Sure.
The FDA had decided back in 2002, when the first recombinant tests were rolled out, that it was not going to regulate them as a blood product because they're not. They're recombinant. They're produced in a bioreactor. This really concerned people on the microbiology committee who had spent a long time seeing how variable this blood product can be. And they worried that there would not be the same sort of good manufacturing process standards applied to the recombinant test as the existing sort of incumbent tests. The other thing I think that happens here, though, is that because of the FDA regulation, there's a pretty small number of companies that are allowed to make horseshoe crab blood-based endotoxin tests in the US, whereas a recombinant test that doesn't have that regulation, in principle, anybody could make. And I think that both poses a commercial threat to the companies that are selling horseshoe crab endotoxin tests, but also raised doubts for people on this committee who have sort of said, well, gosh, anybody could put anything in a vial and call it recombinant factor C. Like, how would we control against that? In principle, that would be something you could tell by doing sort of the standard positive and negative controls that are built into every pharmaceutical manufacturing facility's processes. Like, whenever you get a new test, you have to demonstrate to the FDA that it works to do what you want it to do. But the committee was still very concerned about this.
That led to some serious drama, if I remember from your story.
You could call it that. Yeah. This committee of expert microbiologists, many of whom had volunteered for the U.S. Pharmacopoeia for more than a decade, and many of whom remembered the long process of switching from rabbit pyrogen testing back in the 70s and 80s over to horseshoe crab-based endotoxin testing, which is sort of used mostly now. The USP dismissed a whole microbiology committee because it said they just couldn't work together, couldn't get along. And that was the subject of, I think, a lot of interest and a lot of speculation, kind of from across the industry.
Yeah, I think the natural question that's also very hard to answer is, were they dismissed because they didn't have a good working relationship, as Pharmacopoeia said, or because they weren't coming to the conclusions that people wanted them to come to?
That the Pharmacopoeia wanted, yeah. They say, the people who were on the committee, I spoke to 5 of them out of the 14 that were dismissed, they say it's because they weren't coming to this politically desirable conclusion that the Pharmacopoeia management wanted them to come to, which would be to authorize the use of recombinant tests on kind of the same footing as the horseshoe crab-derived tests. The Pharmacopoeia says that it's because the group dynamics in this committee made it impossible to make progress. And indeed, in speaking to some of the people on the committee, there were times that committee members were lecturing FDA liaisons to the committee about why they were wrong about stuff. There were times that the committee meeting proceeded without ever getting to stuff that was pretty important that they needed to be discussing because they were stuck on this endotoxin issue. And certainly even today, a few years after all of this drama has gone down, many of them are still willing to spend an hour on the phone with a journalist arguing for reasons that recombinant endotoxin test is less preferable to the existing horseshoe crab-based endotoxin tests, and we need more data to be sure that it works the same.
Interesting.
So I think, to me, that's one of the most interesting things about this story is that question of like group psychology and group dynamics and what it takes to be sure of something in a scientific sense in a setting where there are also all these other non-scientific reasons to weigh one side or the other side.
So what do you think is the future for horseshoe crab blood in the farmer world? Is it on the way out, or is there still more fighting left to be fought?
I think it's really hard to predict right now. I think that the environmentalists are kind of gearing up to hold pharma companies accountable. I think thinking back to Tim and that meeting that he told me about, it sounds like among pharma companies, as among the USP Microbiology Committee, there remains still some question about which type of recombinant reagent to use, whether the recombinant factor C alone or the recombinant cascade reagent. And the other thing I think that we haven't really talked about, but that is significant, is even if everyone decided every new drug that we are going to put on the market is going to be tested only with the recombinant reagents, even so, that leaves the entire pharmacy worth of existing drugs that are approved by the FDA or the EMA or various other international regulatory agencies with the stipulation that they be tested for endotoxins using horseshoe crab reagents. And unless or until there's further regulatory change to make the recombinant reagents equivalent to the horseshoe crab reagents, that those drugs that are already approved will need to continue to be tested for as long as they're sold. Until that sort of tranche of regulatory filings expires, which is expected to take about 20 years, there will still be a demand for horseshoe crab-based tests.
Because the drugs are not just approved as a substance, they're also approved as a manufacturing process for that substance.
Yeah. When you approve a drug, it's such a highly regulated industry. It's really interesting. You don't just approve the end product molecule itself. You approve how it's made, where it's made, the facilities, the processes, the protocols, the testing, like we talked about before, of all of the quality tests to make sure that the quality tests are high quality enough. It's there's a lot of layers of consumer protection.
So, Laurel, thank you for diving deep with us on this.
Thank you, Craig, so much for having me. It's really fun to tour the floor of the Delaware Bay with you.
Yeah, next time I go out there, I'm going to be a more informed visitor. So listeners can find me on social media as @CraigofWaffles. How can they get in touch with you?
People can find me as @Laurel Oldach on Blue Sky or on LinkedIn, where I'm under my name.
So you can find Laurel's cover story about horseshoe crab blood on C&EN's website or in the October 28th print issue of C&EN. We put a link in the show notes, along with the episode credits. We'd love to know what you think of C&EN Uncovered. You can share your feedback with us by emailing cenfeedback@acs.org. This has been C&EN Uncovered, a series from C&EN Stereochemistry. Stereochemistry is the official podcast of Chemical and Engineering News. Chemical and Engineering News is an independent news outlet published by the American Chemical Society. Thanks for listening.